Sudarshan Chari from Ian's lab. I'm a fifth year graduate student. Been doing this project for 2 to 3 years actually now. I'm in Ian's lab and my primary role is to look at flies. When they are unconscious, I dissect them and look at their wings. And when they are alive, I look at them have sex all day. So this project is my observation based on those things.
They have wings and have sex.
So to start out, populations are frequently exposed to deleterious mutations, and these can cause fitness defects and consequently a fitness decline. If you direct your attention to this panel, this is a wild-type Drosophila wing, and the other 4 wings, as you can see, have visible phenotypic defects. These are actually wild-caught flies that were inbred in the lab. About 40% of those have these defects. And selection tends to be pretty good at eliminating harmful mutations. But there can be oftentimes where certain situations arise where these mutations can either rise to high frequencies or even get fixed in populations. In this classic experiment, Birch and Chow passaged phages through serial bottlenecks, and as you can see, about 20 generations later, there was a drastic fitness decline consistent with fixation of these mutations. Another study, when Chun and Fei were looking at frequency of mutations or alleles in hitchhiking versus non-hitchhiking regions, they found that disease-associated alleles tend to occur more frequently associated with hitchhiking regions. So hitchhiking with adaptive alleles can also cause fixation of deleterious mutations. This charismatic organism here, charming one, is a pest of cattle. It's the blowfly. And in this study, the authors observed that when these flies were given pesticides, they quickly got resistant to those. And this has a plot of resistant genotype versus the mean asymmetry, which is a measure of fitness here. And you can see that whenever the resistant phenotype occurs in the ancestral or plus population, the asymmetry is higher, indicative of antagonistic pleiotropy. So you have deleterious mutations that get fixed in a population. What are the possible evolutionary strategies? And one of them is amelioration of fitness by compensatory mutations. Oops. And what are compensatory mutations? You have a harmful mutation A. There are second site mutations which arise, which can ameliorate or reduce the fitness cost associated with the original bad mutation. It is conditional only upon the original mutation. By themselves, compensatory mutations are either deleterious or neutral. Okay? A quick example. This organism is Dictyostelium. When these authors fixed a mutation that ablated the swarming motility.
It's actually myxobacteria.
Oh, sorry. I'm sorry. Myxococcus xanthus. I'm extremely sorry. I'm nervous, sorry. These bacteria tend to aggregate and have a swarming motility, and ablating that is detrimental to fitness. When these bacteria when the ancestors were fixed with mutations that affected this motility and allowed it to evolve, they found certain strains which fixed for compensatory mutations and actually compensated for fitness in this case. A quick overview of how these studies are typically done. So you have a population and you introduce repeated bottlenecks and recover individuals which are mutated. And then you let them expand in population and recover fitness. This is a typical mutation accumulation study. Okay? Or you can introduce fixed or known mutations into an organism and get mutated individuals and let them expand and recover fitness. A few general trends. You have compensatory mutations that can arise for initial mutations of either low or either small or large effects, and these can have varying effect sizes. So what is the model that I'm trying to get at, right? So you have, this is the phenotypic space, fitness space. You have a three-dimensional trait or a trait in 3 dimensions, however you want to talk about it. And these lines here are the measures of that, quantitative measures of those traits which define the fitness. For the moment, just assume this is how the traits should be distributed for a wild-type phenotype, right? There's a mutation which arises in one of the traits, causing a fitness decline. Now, there are 2 options. Either that trait can go back to normal, in which case this would climb up in a similar manner that it came down, or you can still have that, or have other traits evolving to compensate for that, in which case you have a separate peak, or it climbs up in a different manner. Similar to a rock climbing thing, if you have a peak, you can come down a way, you can go up the same way, or you can take an alternative route. Anyone ventures to guess what this rock is? Rock formation is? It's Beacon Rock. It's in Washington State. So many ways to do well in Beacon, eh? So coming back to the presentation, what are the main questions? So you have this situation where you can have a deleterious mutation affecting a trait in some assumed focal observed trait, okay, or a bunch of traits. How does evolution get the fitness back up? Is it by affecting that same trait in a similar way and taking that trait up back, or it's by evolution in other traits? How important is standing genetic variation in providing these effects? Now, for a sexual diploid organism, you might have mutations which are already present in a population, which can buffer traits, which can buffer harmful mutations. How important are these, or is it just all new mutations that we are waiting for? And what is the genetic basis? What are the genes? How do developmental organisms circumvent developmental problems? How many genes? What are their effect sizes? Small effects, large effects? How does the combination affect these dynamics, etc., etc.? So our study system is Drosophila melanogaster. Female, male adults lay eggs, 3 larval instars, pupate, and back to adulthood, metamorphose into adults. Okay? Skipping over that. And quick developmental overview. The adult structures are generated by these bags of cells called imaginary discs, and we would be focusing on the wing imaginary disc, or the wings basically in adults. That was Drosophila development in one slide, right? So, and we know that we are looking at wing because we know that it can affect different components of fitness. The obvious one is flight performance, right? But in the lab, it may not be as important to look at flight performance. I don't know, I'm assuming this. But what is more relevant to us is this courtship behavior. Now flies use these wing displays for courtship, and they use it saying, hey, I'm here. Also, they vibrate it and produce a song. So it's both visual for the female as well as the vibration frequency, so an auditory component to it. So both, okay? And also male aggression behavior. Now I am going to show you a tiny clip. That's a male fly approaching a female, and that's the wing display. If you notice, the female is decapitated. So it's a pretty robust behavior. It doesn't even so I am not showing what happened next. Oh, there was another male, and they tried to mount the female from both sides, the decapitated side and the actual side. I just did not show that here. So wings are important, and this component of sexual signaling is pretty robust. And there's direct evidence for this, right? If you amputate the wings, as shown by cuts here, here, and here, there's a linear decrease or increase the way you look at it in the success, in the mating success and hence mate acquisition. So wing size and wing area matter. In addition, our lab has also identified the sexy vector. So Will can talk to you about how he did it. So if a fly, so when you correct the size of the wing, if you have a shape of the wing which looks like this, you don't want to be there. Females don't like you. They choose you less than if you have wings that look like that. So you have that fine scale resolution of the mate song which the females can detect. So presumably wings can be a target of selection even at a tiny, tiny level. What do we know about compensation of wings? In an experiment done way back in 1989, it's probably 5 years old, these authors introduced a mutation called vestigial in the wild-type fly. This is a wild-type thing, and that's what the mutation does to the wing. It destroys the wing, basically. Okay, and did artificial selection, selecting for wild-type things every generation. And what they found was, in about 40 generations, they were able to recover almost wild-type wings. But we have no idea about the fitness in this case, or we have very little. We don't know what happened to the fitness of these flies. On the flip side, the Shedder and Chippendale introduced a completely different and independent mutation, nubbin, into the wild-type wing, into the wild-type fly, and recovered components of fitness in Drosophila by natural selection. That's relative male fertility in evolved populations versus the ancestral populations. But in this case, they don't mention anything about the phenotype. Also, this was just done once, there was no replication to it. So what we decided to do was study this. And again, the reason why we are looking into this is that causal link between phenotype and fitness, whether it's compensation by that focal trait or by evolution and other traits. And so our study protocol basically involved fixing of a bunch of deleterious wing mutations into a large outbred population and generating 3 treatments. First you have the control, then you have the unevolved mutant, the generation 0 before we started any selection experiment, artificial selection treatment, and a natural selection treatment. The phenotypic effects of the mutations used were, it was the same vestigial mutation and another nubbin mutation. Now these two are developmental mutations, strong developmental effects. We also used a bunch of weak wing patterning mutations. Now these mutations, when the flies are wild caught and when they are inbred in the lab, they uncover mutations which have phenotypes pretty similar to these. So, and a bunch of studies using complementation tests have demonstrated that these genes are actually the ones causing these mutations. Not these exact mutations, but alleles in these genes. So we are pretty much fine using these mutations. For the purposes of this talk, I'm going to talk about just this mutation. That's what I've quantified. So that's all I've got. A quick overview of what this can do. It ablates the wing, so it affects mate choice in the adults. It is also known to have pleiotropic effects in the larval stages. In terms of developmental time, probably competitive ability is kind of correlated, and viability. So lay an egg, how many adults it grows, basically. It affects that. So the first evidence that I provide where these two play a role is when we have these mutations along with wild-type flies, the frequency rapidly goes down. Okay, and this is more so with mate choice, and less severe with no mate choice. So you have a non-sexual component here, and you have a sexual component here. So both these effects are playing a role in mediating this loss of this allele. This is done by Cody, an undergraduate. So what are the selection genes? I talked about natural and artificial selection. Natural selection cages look like these. In a population cage, we dump about 3,000 flies, extremely high density, and we let them lay eggs in bottles for about 5 days or 6 days, so very high egg and larval density. Those are actually flies sitting on the top of the bottle, and this, you see, actually is the egg mass. That's egg mass within 1 day. It's quite high. And I'm not selecting directly on the wing here. There is no artificial selection on the wing. So we just let them do their things. And then there's an artificial selection replicate where the adult density is low and the larval density is also low. And here I artificially select for 55 most wild type looking wings every generation, and they see the population for the next generation basically. So two different. What we see here is in the artificial selection flies, wings look like this when we started out. In 14 generations, we see an immediate difference. So there's a rapid increase in wing size, and it appears to be dimorphic. These are males. Males compensate quicker than females at this point at least. But there is no change in the wing phenotype in the natural selection treatment. So, and these results in the final phenotype are mirrored in the cellular phenotype. So you have a wild type looking imaginary disc. This mutation is supposed to cause cell death, which is by death. In artificial selection flies, cell death is rescued, not in natural selection flies here. So in this case, if there's a fitness recovery, it's not due to the wing because the wings are not changing. So severe reduction in wing size leads to a loss of these sexual signaling components. So there's behavioral modification now, some sort of behavioral yes.
Sorry, I thought you said that the natural selection was totally ineffectual, and I thought this is now saying how did they do it with natural selection?
Yeah.
Or is this just hypothetical?
Yeah, it's if they have evolved, it's not by wings. So we looked at behavioral modifications, and that's the part which is relevant for today. So have we seen behavioral modifications? Yes, we have. When new males have evolved or arisen in certain Hawaiian islands, it's known that females respond more to the chirps as well as discriminate less against whom she mates with. There are other modifications that the males have done which also plays a role in this. Similarly, in wolf spiders, for the courtship ritual, they drum this front feet and wave these pedipalps. But in nature, there are often times when these feet get broken off or are eaten off. So they compensate by waving this at a higher intensity. So there is some behavioral component or modification when things happen. Again, why did we expect this? Because we knew that wing size was a player, and now wing size and shape are not changing. So is there any other way? Again, coming back to this fitness effect, we looked at mate choice. Did the mating behavior itself change? So to test for behavioral compensation in this case, we had a population cage in which we had 15 males and 15 females from either wild type, natural selection, unevolved mutant, and artificial selection, and looked at the proportion of males copulating and or courting of every 5 minutes for a total of 70 minutes. Okay? And what do we see is that the proportion of natural selection males that copulate with females is slightly higher. There is a trend there. And it seems to be that they are courting more vigorously. Probably there is a correlation between that. So they seem to be persistent. So we directly tested for this and looked at, if I gave males from the natural selection population, females from all the other populations, how did they respond? Did they change their behavior? Did they modify? And conversely, we did the reciprocal. If we gave females from the natural selection population, males from other populations, how did they respond? And we did the same thing. We looked at this population and courtship in a male biased manner. So, if I used the male and crossed it to a wild type female, is the same, almost similar to this, as if I crossed it to artificial or natural selection or unevolved. So the males are consistently courting at some rate. Okay? This is proportion of number of flies courting. But the reciprocal is not always true. So if you give a wild type male a natural selection female, the courtship is not that big. Same goes, but it's different for these guys. For the base, again, it's long. So in this case, the males are consistently courting in a high manner, or they're very persistent. But does that lead to success? Probably not. There's a variable success, but the take-home message here is that in the case where you give them unevolved mutations, they just don't like it. So high courtship, variable success, especially so when it's the unevolved mutation. So they're doing something better. So now we know that there is some change in the behavior. So we looked at these other effects. So we looked at viability, and we did it in two ways. First, we gave them 8 to 10 ml food, 50 eggs, perfect conditions for growth, no stress at all. And then we let them grow in high densities. Now remember, most of these populations are grown at high densities, so it is their evolution regime basically. And we looked at how many flies, when we introduced the eggs, how many adults did we recover at the end of the hatching period or eclosion period. We found that at low density, there's no change. Almost all of them do fine and hatch. But at high density, there's a difference. So, just to illustrate that, the wild type are the best, followed by the natural selection, followed by the unevolved mutation. I have not depicted what the artificial selection did here, and if Ian does not mind, I did a quick, he does, so I'm not going to say anything. So, I did a quick number calculation of how many flies I got, and the artificial selection flies have fitness which is quite comparable to unevolved flies. So they are evolving in terms of the wing phenotype. Yes, we are selecting on the wings here, and they are also recovering fitness when you look at the mating behavior. But in terms of life history, not much. They are not doing much. So again, a rapid wing recovery in artificial selection. Remember those other 3 characters I talked about? These mutations. We seem to be getting similar reactions here also. So wings are recovering via artificial selection. But there is no change in the wing when we look at natural selection here. But at least for this particular mutation, there is behavioral and life history compensation of some sort. And so, going back to the model, what seems to have happened here is not that, but that. And I'm sorry for the awful covers. And a bunch of future directions. We are going to do a bunch of choice assays next and look at actually the female versus male mediated changes in behavior, testing female fecundity and competition for more life history assays. And then, this is in progress, I'm trying to extract DNA from these evolved flies and look at underlying changes that are responsible for at least the artificial selection in a developmental sense for what the compensation here. And that's my lab. Thank you all of you. Thanks, Deacon. And I didn't have a picture, and she is the undergraduate from heaven. Font 44 for her. Yeah. Thank you.
So it's really clear results. It's cool. It fits this difference of the artificial and natural selection. Do you think you would have gotten different results if you had, say, seeded the populations with a very low frequency of the wild type? In other words, given them, so you started, I guess, with the fixed inbred line with the mutations. So
No, this was outbred. So we collected flies from...
It had the heterogeneity, but at the locus that was manipulated, it was homozygous.
Yes, yes.
If you had seeded a very low frequency, I wonder whether, like in the high density condition, they might have just climbed, whether the easiest route, had there been a revertant present, would have been to go back up. Can you do these experiments?
He's done it. We already did.
Cool, excellent, yeah.
So,
It's a nice little control then.
We lose the
You do lose it.
Yep. So this is, so although I've
Nice.
plotted the vestigial homozygote here, the vestigial allele is at 70% frequency here. So, and there's a rapid loss within 4 generations. So we had like 8 out of 200 flies, only 18 were wild type flies, and even at that frequency, there's a rapid reduction and loss of these alleles. This mutant phenotype is also.
I see, so they do just go straight back.
Yep, yep.
Even in the even in the
So this was done at
natural selection kind of thing, where it was so crowded and there wasn't much water.
So this was a separate experiment. We were just testing how quickly does this allele go extinct, so to say. And this was done at about a population size of about 200. And that was, so even at that, it's going down. So I don't know whether it directly answers your question, but I think if you have a wild type, it's infinitely better in this situation.
Yeah.
No matter what
It's
the
just
start.
too big
Yeah.
of a difference.
Yeah. Anyway, go ahead.
The wild types were always males, right? What if they're female?
They will, in what sense? No, this is a mixed population. So there's a mixture of males and females of all genotypes.
Okay. And so there was a mixture of
Yes,
wild types?
yes.
Okay.
In your artificial selection, you said the cage was very sparse. I guess there wasn't a whole lot of
Yeah.
flies in there. Do you think that might affect, or might be some of the reason why when you then took those guys and put them in a high density
Yep.
population,
Yep.
they were terrible?
Yep. We made sure that we had a different selection criteria. We did not want natural selection, so to speak, to do much. I'm not saying it did not, but in this case, I was the selector. I would go and pick out the best flies, what I thought was the best flies. Yeah.
So is it just a physical limitation that you can't have 3,000 flies
Yes.
on the right side?
So
I figured as much, but you know.
yeah, I would probably kill myself.
Well, so, and this is, the low density is actually doing the flies a favour from
Yeah.
the point of view of natural selection, right? This is,
Yes. Yes.
So in the natural selection experiment, you didn't necessarily see compensatory phenotype or wing shape, but you did see compensatory developmental and behavioral
Yes.
factors. Are those due to mutation, de novo mutations, variation, or
So we tested those. We tested everything within 25 generations. although I've put up pictures of about 15 or 18 generations. It's too rapid, at least for the phenotype, so I'm not ruling out new mutations, but it's predominantly possibly due to standard genetic variation. That's my least controversial answer to you. Flies are crazy, I don't know what they do, but according to theory, at least I believe it's standard, the extent variation present in the population.
Why did you decide to make the natural selection group a high density group?
Because the flies which we got in nature, or which we collected from the wild, were at extremely high densities, and it's not an unreasonable argument that this is what they face outside nature. We don't have.
Just that there's another question over there when you get a chance. Yeah.
She asked first, so I'm going to go with her. That's fine.
So I'm just wondering if you had started off your natural selection population not with the wing mutants, but just normal wild type flies, if you would have seen the same increase in the level of competitive ability, like whether that's really compensating for the wing sort of detrimental, or whether it's just adversarially compensating.
So there is evidence that if you put flies in high density conditions, they do compensate for these life history traits. But in this case, it gets complicated by the fact that they have the mutations in them. So it's an interaction between both selection and how sick you are at that point. So definitely yes, but then does that fitness also somehow manage to increase the wing phenotype?
Yeah, I'm just wondering whether you can really call it compensatory if it might have been an axis that would have been adapting anyways.
So the more interesting part in this case is, as adults, even if they did compensate, or even if they did not compensate as adults, they can still increase their persistence or behavior. So it's not as if they're not compensating as adults and are only compensating in the pleiotropic or the other life history traits. They're doing both, a bit of both. And they have not, even though they're compensating for it, they have not gone to the level of wild types yet. So there is definitely some effect of that mutation still acting on these flies. So it's like a combination, if I might suggest.
They had a question here.
Question?
Can you hear me?
Yes.
Okay. So I see you work with a wide population, 3,000 flies.
Yes.
I was wondering, do you have any reason to believe that the environment may not have an impact on that compensation? Because you talked about the shape of the wing, the size of the wing, but what about the environment, such as the temperature at which you kept your flies or the density of the air, things like that? Do you have any reason to believe that they wouldn't have impacted that compensation?
They would have, but I kept the temperature constant, so they were all reared at the same temperature at very high similar densities. So whether environment has played a role, probably yes. If I had looked for, say, tolerance to bad food, they might do well. But so in short, yes, environment does play a role, but I've tried to keep it constant with respect to temperature and rearing density within each treatment. Does that answer your question? I've tried to control for it in some sense.
I think it was a slightly different question of if you reared the experimental evolution flies with the small wings under low density conditions, would they still have little wings? So are you just looking at plasticity in the wing?
My guess is no. I don't think that would happen. It's not plasticity per se. There is definitely an effect of environment. So if you rear flies at lower environment, as a whole, the fly looks small or big depending on the temperature, but that does not mediate this evolutionary response because it's hereditary in some sense. I see this fly getting larger wings every generation, no matter the temperature in this case. Does that make sense? So it's not environmentally determined, it's genetically determined in some sense. I don't know.
So you say it changed no matter the temperature, but your experiment were carried at one constant temperature,
Yes,
right?
yes. The rate might have been different if I had done it in different temperatures, but again, it would be logistically difficult.
Right.
That's all I can say. I have not done it. I had thought of doing it,
Okay.
but not possible. Yes.
So, this is sort of a strange question, but would it be two parts? Would it be possible to artificially select for the life history changes and the behavioral changes in the same way you did wings? And then, if that were possible, is there a way you could test sort of fitness test that would be comparable between the three of them?
So there is a way. What I could have done is made wing clips every generation and looked for how they were compensating for behavior. So this is just at the behavioral level. At the same time, I could have, again, taken the wild type and grown them at extremely high densities and just looked at how it changed. But then it would be very difficult, and the comparison is still not direct at that point because you're clipping it post being born. So they still have that mutation. At that point, they would just learn to either behave, either be more persistent or accept more. And that it would be a learn. So I don't exactly know how to tease apart in that scenario that way. It would be difficult, let's put it that way. I can only say that.